So this is a small introduction to what we're going to discuss in class. And here we're going to talk about the B lymphocyte development in the class. In this video, you can see some of the steps that we're going to discuss here. They are talking in more detail. I'm going to, as I only have half an hour, I'm going to give you an overview of the process. And in this lecture, I'm just going to give you the basics, things that I want to make sure that you know. I'm sure that you know them. You can skip the video, or if you have any doubt, you can watch the entire video. So let's start. So now I'm going to tell you what I'm going to talk. So in these 10 minutes, I'm going to tell you that in the same way that humans have created weapons and those weapons have evolved, they are constantly updating. Our immune system is doing the same thing. So for example, during lymphocyte development, you will see that the receptors, the B cell receptors in this area where the antigen is uh, going to attach are constantly updating. And the same thing will happen with the immunoglobulin, that this area where the antibody is going to attach to the antigen is constantly changing. And how this change? It changed by several genetic mechanisms that I'm just going to talk very briefly. I just want that you remember the names. But all of them work in a relatively similar way. That is, you have few genes, few pieces like the Legos, and you can create a huge combination of shapes just by interchanging them. Yeah, so maybe you have heard about this, that is very famous, the BDJ rearrangement. If you see here, this area that we just talked here with the receptors and the immunoglobulins, both, if you see all these different colors, represent that there is a variation between the potential genes that are located there. And something similar, this rearrangement can also happen in process like somatic hypermutation and the isotope switching. Okay, this is just an introduction, so I just want that you remember that this is the topic that we're going to talk. So I'm sure that you know what is a B lymphocyte. I'm sure that you know it is a type of blood cell that is very important uh, in the adapted immune system. So important features of the immune system like the specificity and the memory is in part due to the B lymphocyte and in part due to the development. So I'm going to try to explain in the lecture how this happened. And these are some of the key roles of the lymphocytes B. So the production of the antibodies, the immune memory, and the immune response. So for example, in the immune response, you know that the antibodies are going to neutralize, are going to be blocking, yeah, attaching to the pathogens, and then they will trigger an immune response that our cell is going to go and kill that cell. So for that reason, understanding the B cell development is very important. So for example, the immune memory, yeah, is very important for the vaccination. So now that we understand that we're going to understand this better, we can understand much better all these important processes that happen in this adapted immune system. And I'm sure that you also talk about this arm race between the pathogens and the immune system, and that the human civilization is always updating their weapons. Same thing is happening with the immune system and the pathogen. So every time that a bacteria and a virus had a change, our immune system has to find new ways to detect them. So for example, in our case, the B cell receptor need to be changing all the time, yeah, to attach to the antigen. Yeah, so we have to find a way to attach to the new part of the protein that is changing. And after that, it also have to attach to label it. Yeah, so same thing that happened with your antivirus in your computer. If you have a virus in your computer, first thing searching it, then the antivirus say, like, could we destroy it? So same thing happened. So you produce antibodies that are going to label it to later destroy it. So remember, uh, why is important to understand the B lymphocyte development, because this is how you detect the antigen. So you're going to find these very important proteins that are the B cell receptors. Very important to search for the virus, and they work. I'm sure that you know this with this lock key mechanism, that they are just using complementarity, yeah, physical complementarity. So they need to, like the lock and key, the antigen and the binding receptor need to be changing to match perfectly. In the same way, the antibodies, and I'm sure that you know this, they are able to or neutralize or they are able to mark for removal. So, for example, the immunoglobulins are going to say, let's mark this. This is a bad guy. Now another cell can go and kill it. And this use also the same mechanism of the lock and the key. So, so both are just changing to be complementary to the antigen, to the new mutation in the virus or in the bacteria. And also the antibodies can neutralize, so they can surround it, so the bacteria or the virus cannot attach to the cell anymore. I'm sure that you know about this. If you have any questions, you can ask me later. I just want to make sure that you remember all those things. And you have here maybe before about these some genetic mechanisms involving this rapid development, rapid evolution. So one of them is the BDJ rearrangement. I send you some notes that if you want you can read before class. And here you can see that I will explain this in a little bit more detail in class, that here this is the region on the top that have a different colors. This region is where the receptor they had to attach to the antigen. So here is where the cell receptor is going to attach to the virus or the bacteria, and this is the one that has to vary a lot. So same thing that happened with the Lego. You have different type of genes. You choose some of them at random and then mix them there, and this will create a lot of variation that allow them to some of them attach to these new antigens. And this not only happen with the receptors, with the cell receptors, will also happen with the antibodies. So you have something called somatic hypermutation. We will talk about that, how in class, how they create this variation using this enzyme, and also how they are selected to, so the only the ones that bind the better, that have the strongest affinity will attach. And you have other processes like the isotope switching that create a lot of variation to, for example, to have the best immunoglobulin suited to the immune response that you need. Again, we will talk more about this in class, but the key idea here that I want that you remember is the Lego idea, that you have few components, you mix them a little bit, and then you have more variation. So why all this thing that I told you is because I want that you think about this. We only have a little bit more than 20,000 genes, and this is very small. In the beginning, we thought that we had more than 100,000. Why? Because we thought that it was impossible for a human being to have the immune system so condensed, all the responses. And maybe if you watch this old movie called The Matrix, you have heard about these key makers, that this is a key character, an important character that is making all the keys to open some doors. If you want to watch that movie, the idea that our immune system was able to create all the potential keys that open any lock, in this case, that attach to a huge diversity of antigens, yeah, both using the receptors and using also the antibodies. And this then is done, as I said before, by the combinatorial gene segment recombination. And I told you, and we will talk more in class about this BDJ recombination that are happening in the BCR receptors or B cell receptors, the isotype switching, as well as the somatic hypermutation. Okay, I hope that this is clear. Now let's talk a little bit about these B cell receptors and the immunoglobulins. So just a quick introduction, you might know more about them. So what happened during an immune response? There are three things that are happening. So you have the antigen here, that is a bacteria, or in our cases, a virus. The antigen will be any substance that can cause an immune response, that it could be one of the proteins or the sugars that are located in that bacteria or that virus. And in this case, one of the responses that it could happen is that the B lymphocyte is going to detect it. Yeah, that it will be in the receptor. And now they have to produce antibodies, and these antibodies are going to label it to say you have to destroy it or neutralize it, as is happened here. Okay, so Then the fourth thing that happens is that it will be an OR cell that it will come and destroy it. Okay, so every time there is a mutation, we need to change both our searching mechanism and as well our labeling mechanism to be destroyed. So I'm sure that you know this. So the B cell receptor is a membrane-bound protein that is expressed in the surface of B and T lymphocytes. So it has different components. So it has, as usually, this heavy change and this light change. And in addition to that, as is a membrane-bound protein, it will have a transmembrane region. So it will be located here. That it will attach in that area. So remember, the structure is a heavy change and a light change. And remember, you can also divide it by a constant region. Here you can see a constant region, that is the one that is not going to change, and a variable region. And as I said before, why this is a variable region? Because it's the one that it needs to be changing to adapt to the changes in the mutations in the antigen. Yeah, so it attach properly. And what is the function? Is recognize the binding and binding directly to the specific antigens. And as I said before, is highly specific. And how is that? Through the process that I told you, that is the PDJ recombination. So I'm sure that you know about the antibodies and the immunoglobulins. Immuno because it's related to the immune system, and globulin because it's a globulin protein. And if you see the structure very similar, you have here heavy change, you have light change, you have a constant region that change less, and a variable region that change more because it's the one that it binds to the antibody, while the constant region is the one involving the function of the antibody. So remember, what is an immunoglobulin? It's a glycoprotein that works as an antibody in the immune system, and is produced in this case by the B cells and by these specialized cells called the plasma cells that we will talk later. And it's composed of two heavy change and two light change. And of course, it will change through two different mechanisms that I already told you. It could change by the somatic hypermutation, and it could also change through the isotype switching. So we finished this introduction. Hopefully it was useful if you need it. Remember, we talked about this arms race between the immune system and the pathogens, and that for the B lymphocyte development, it's very important to understand that both the B cell receptors and the immunoglobulins are constantly updating to attach to bind to the antigens. And there we have many genetic mechanisms, and I show you this mechanism of few components that organize in different ways that rearrange it, create a lot of diversity. And I told you that there are multiple mechanisms that we will talk in class that they do that to produce more diversity in the immune response during the B lymphocyte development. I'm looking forward to see you in class, and have a good day. Bye.